This is a cell cycle. To begin, we need to talk about the smallest component of the cell cycle, the nucleotides. There are 4 different types of nucleotides in a strand of DNA: adenine, guanine, thymine, and cytosine. Adenine pairs with thymine, and cytosine pairs with guanine. In a strand of DNA, when the nucleotides bond together, the bond holding them together is a hydrogen bond. This means it is not a very strong bond and can be broken easily. The sugar phosphate group is the backbone of the DNA strand. When the nucleotides bond together with the sugar phosphate group, they are held together by covalent bonds. A strand of DNA is like a very long and twisted ladder. During DNA replication, the double helix is unzipped by an enzyme, and 2 new strands of DNA form. DNA polymerase is an enzyme that corrects any errors made with the nucleotides that are paired together. DNA replication is semi-conservative, which means only half of the original strand is left, and a new strand is formed. Chromosomes are like very compact X's that are made of tightly coiled DNA. DNA uses histone proteins to wrap themselves around and make sure the chromosomes don't get tangled, and the DNA stays organized throughout mitosis. They eventually form what are called sister chromatids. Each sister chromatid is half of the X that makes up the chromosome. The sister chromatid connects to another sister chromatid at the centromere to create a chromosome with the X shape that we are familiar with. The first phase of the cell cycle is interphase. During this, we have preliminary growth of the cell, where it carries out its functions, and in the next phase, we have synthesis, where DNA replication occurs in preparation for mitosis. Next phase is gap 2, where we have additional growth before mitosis. Next, we actually have mitosis, which has 5 stages in itself. 5 stages are prophase, metaphase, anaphase, telophase, and cytokinesis. The first phase is prophase, where DNA compacts itself into the chromosomes and the nucleus itself dissolves. In metaphase, the chromosomes line themselves up along the center line of the cell as spindle fibers connect them. The spindle fibers are thread-like structures that are used to pull the chromosomes, and they connect to the centrioles. During anaphase, the spindle fibers pull the chromosomes apart, pulling sister chromatids towards the spindle fibers. The spindle fibers are anchored to centrioles in the cell. In telophase, the last step of mitosis, paired chromosomes are separated by spindle fibers to the poles, or the 2 opposite sides of the cell. After this, the cytoplasm will divide up and prepare for cytokinesis. In cytokinesis, the 2 cells separate, creating 2 new daughter cells with their own nuclei containing identical sets of DNA. Cytokinesis, cell division, is a form of reproduction that is known as asexual reproduction. Asexual reproduction is with only one cell splitting itself to make 2 new cells. Another form of reproduction is sexual reproduction, or reproduction between 2 cells which reproduce to make 1 new cell. Each type of reproduction has its own advantages. For example, asexual reproduction is usually favored in a stable environment that doesn't need very much genetic diversity. Since asexual reproduction doesn't create any new genetic combinations, no changes to the population will be made. However, sexual reproduction is generally favored in changing environments that require genetic diversity. Sexual reproduction mixes the genetic information of 2 different organisms to help create a very genetically diverse population. This is important in a changing environment because new traits and combinations are needed for a species to adapt to and survive any changes in its environment.